Alright, let's go ahead and get started. I wanted to start out today with this case study. So this is a pretty small one. I think actually this kind of reiterates a bit of what we spent some time on in lab yesterday. So we'll start out with this. We'll talk a little bit about the wikis, which overall look really, really good. Everybody ended up doing it, which I am wholly impressed by, so nice job. And then we'll spend some more time talking about gram-positive and gram-negative, some structural differences, and finishing off with some gram staining questions. So those are the major plans for today, and then before you leave, I'll give you your assignment for over the Labor Day weekend. Okay, so I'll give you 5-ish minutes. Work in pairs, 3s, 4s. On this case study. Okay. Anybody not have a copy? Okay. Wasn't added for a long enough period. And yesterday when I was looking around, I didn't see so much instances of that. It was more often over decolorization. And I said this is kind of the most sensitive step. Is gram staining perfect? No. Is it good? Is it a good diagnostic tool? Yes. But we never rely just on gram staining. In fact, in the past probably, 5 to 10 years, there have been a host of new, more molecular techniques that help us to identify microorganisms. Has anybody heard of the term deep sequencing, next-gen sequencing, anything like that? I mean, there's a plethora of places now that will sequence your genome for you for under $1,000. We can do that with bacteria too, real easily. So a follow-up to this might be some metabolic tests, might even be to sequence everything in that cerebral spinal fluid to determine what's there and what shouldn't be there. And we'll talk about that actually quite a bit later on in the semester. Gram-positive should be what color? Purple, gram-negatives, pink, red, somewhere in there. Acid-fast. They should be like so acid-fast are a little bit different, right? Acid-fast might go back to, that's not on this here, but it was in the table. Acid-fast are one of the other types that I wanted you to be familiar with. I'm going to flip ahead to that real quick. We'll talk about why they are different. So here's an image of an acid fast. And in the acid fast bacteria, we have cell membrane, just like we find in gram-positives, peptidoglycan, just like we find in gram-positives and gram-negatives. What's unique about the acid fast, Derek?
The mycolic acid.
Yeah, the mycolic acid, which it gives us the name Mycobacterium. Okay, so this is, and the consistency of this acid is waxy. At room temperature, it's a waxy-like substance. It actually makes it very challenging for white blood cells to phagocytize Mycobacterium in the same way that we talked about the capsule on Streptococcus makes it challenging. And because this is waxy, it's a solid at the room temperature, a little bit above room temperature. Those dyes that we used yesterday, they can't penetrate this mycolic acid. And so what we find then is the way to treat these, and Derek, I think you were one that actually answered this question on how to stain them. How do we stain acid-fast bacteria?
You have to heat it to liquefy the acid-fast structure in order to set the stain.
Good. And usually the stain we use is called carbolfuchsin. I'm not super concerned with you knowing that, but you may find it. So it's spelled carbolfuchsin. And it's just a fun word to say. So we heat them up, we add the dye, and then we snap cool it and drop the temperature pretty quickly. That re-solidifies that waxy layer and traps the dye inside of the cells. So we could classify these as being part of the gram-positive because one of the things that they don't have is an outer membrane in the way that gram-negatives do. But usually we kind of classify them as their own group. They're kind of a specialized gram-positive. Okay, so we've kind of answered the first and the second question in some ways, and we've answered the third one, so we've actually gone through this whole case study. Right. What I wanted to do is to address a couple of things that came up in the tables that, or in the wiki tables. One is on sporulation. There was a common thing there that I want to address. And then the other one was, one of the things that I like about how this class is running this semester is I learn a heck of a lot of stuff too. So if there's one fact, and I don't remember who provided it, that I'm gonna ask who it is later on, because I need to find the source for that. And then there's something that I said yesterday that I had to go back and I figured out I was wrong. So I'm gonna clarify that as well. So the first one, and we'll talk about this more on Wednesday next week, is sporulation. So endospores form inside a bacterial cell. The spores are non-active. They are not actively growing. And usually what we find with spores is that they have a really thick coat that helps for them to resist a variety of different conditions. They're heat tolerant, they are cold tolerant, they're acid tolerant, they are extremely tolerant to alcohol. Alcohol has no effect on them whatsoever. Resistant to UV stress, etc., etc. And so there have been researchers who have actually isolated spores that are 30,000 years old, and they can get them to regrow in a laboratory setting. So these can survive very, very long times. There's only one group that actually sporulates, and I'm trying to check on this with a friend of mine from St. Norbert to figure out if I'm wrong on it, but to my knowledge, the only ones that form spores are the gram-positives. Gram-negatives form what we call persister cells. Those are not technically spores because they are still vegetative, they're still growing, but spores are only limited to a few genera of the gram-positives. The one that we think of most typically, actually there's two really good examples. Bacillus, the genus Bacillus, so Bacillus megaterium, Subtilis, anthrax, Bacillus anthracis is a sporulator. And inhaling those spores is what leads to the worst disease in the case of anthrax. And then Clostridium, Clostridium tetani, Clostridium botulinum, Clostridium difficile, which I'm going to guess that like over 2/3 of the population in this classroom knows or has heard a personal story about C. diff infections. So those are kind of the two prototypical classes that we think of in terms of sporulation. Staining spores is a little bit different because of that thick coat of theirs. They are also pretty resistant to taking up stain. And so to stain spores, what we typically do is we steam heat them. Basically get a bucket of water, lay a metal grate over the top and put your slide on there. And then you, with that slide, you put on a piece of paper, soak it with a dye known as malachite green, and let it heat for 5-10 minutes, let it cool, and then you counter stain the rest of the cell body with safranin. And so in those cases, what you will occasionally find are endospores that have been released from the mother cell. They kind of end up looking like they look like rugbies more than footballs. And then sometimes you can actually see these spores inside the mother cell. And you don't always need to do the full staining to see that. But Clostridium, for example, kind of form these club-like patterns where that spore is still inside the mother cell. Bacillus, on the other hand, usually that spore is kind of centrally located. And you may see this at some point in the lab. So, all right. So just wanted to point that out. Let's go to the cellular level. I've got gram-positive and gram-negative up here for us to compare. What are some, let's just start with a list of what are some differences between these two. And this kind of harkens back to the table. Ashley.
Gram-positive have a thicker peptidoglycan.
Good. Thicker cell wall, thicker peptidoglycan. So somebody, and please fess up because this is great information, it makes up 50 to 90% of the cell envelope. Did somebody say that in their wiki? No? Damn it. Okay, somebody did. I'm going to find out who it was. In gram-negatives, it only makes up 10%. So that gives you an idea of how thick these actually are. I think that's if you're a mathematical thinker, I think that's a great statistic. But much thicker. We think of the peptidoglycan in gram-positives being multiple layers thick and gram-negatives being 1 to 3 layers thick. What else? What are some other differences? Haley. Good. They have a cell membrane, and then they've got this outer membrane here as well. What does this outer membrane have on it? A couple of features. Meg?
It has the porin inside.
Yeah, and there's multiple different types of porins. This kind of gives the impression that there's only one. There are many different types, and each one allows for a subset of molecules, hydrophilic ones in particular, to move passively or facilitatedly through that outer membrane. And usually, most of these have the prefix OMP, OMP, for outer membrane protein, but some have different names than that. What's another key characteristic of this outer membrane? Yeah, lipopolysaccharide, or we abbreviate it LPS. So that LPS, couple of things about it. From a medical perspective, or from clinical perspective, it can be used as a way to distinguish bacterial strains from each other. So for example, has everyone, I've at least brought it up, but 0157 in terms of E. coli, sometimes you'll see that prefix of 0157H7 or O26. The O refers to part of the lipopolysaccharide. So the LPS has 3 distinct parts to it. It's got a lipid part, which is called lipid A. So that's lipid A. It's got a core polysaccharide. I'll just kind of draw it as a string here. And then all of them have, well, most of them, have also an O polysaccharide. And that O polysaccharide is unique to different substrains of bacteria. So for example, in E. coli, there's O1 all the way through O157. And in laboratory settings, we can create antibodies that will recognize any of those species as a way to distinguish between these individual substrains. Now, importance in terms of the organism, this LPS seems to be required for life. We don't exactly know why, but that LPS, if you delete all of the genes that produce this LPS, cells will not survive very well. They won't grow well. Okay, so that's something that, again, we don't know why right now. We'll come back to the LPS. It's actually also a toxin, and that's something we'll talk about way toward the end of the semester. Other unique features. Ashley?
Periplasmic space.
Yeah, so we've got that periplasmic space. This picture here divides it between 2 parts. I just draw the whole thing as periplasmic space. Some metabolic enzymes in there, some other enzymes that are going to be breaking down nutrients. We will find, and then we've also got these lipoproteins helping to hold that cell wall in between the membranes. We also will find on this inner membrane at some point, we will talk about our electron transport chain. Yay! That's where it occurs in bacterial cells, is on this inner cell membrane. In the case of gram-positives, one other distinctive feature is the presence of these teichoic acids and lipoteichoic acids. The difference between them is where do they originate from. So teichoic acid begins in the peptidoglycan. The lipoteichoic acid originates from the membrane. Again, I should say that as far as I'm aware, no one knows what the actual function of the teichoic acid is. But again, if you delete the genes that produce teichoic acid, gram-positive cells don't do very well either. So it does seem also to be required for growth and for life of the organism. But how? Beats me. Not really sure right now. Given these structures, what contributes to the difference in staining? Why do gram-positive bacteria stain purple, and why do gram-negative stain pink? Derek?
Gram-positive, the peptidoglycan is right at the surface.
Okay.
Stain easier. While gram-negative, the outer membrane prevents the stain from reaching the peptidoglycan, so you have to remove it to stain it a bit different.
Okay. Anybody have anything different than that? Okay, here's where I made a mistake. First part's absolutely correct. I actually found out something about crystal violet, and I should have probably known this. It's actually quite hydrophobic. So, it actually does make it through the outer membrane. So what I said yesterday was that outer membrane prevents it from getting through. And I don't think that's actually true. I looked at a couple of reliable sources, and what I found was that the crystal violet will get into the peptidoglycan of gram-negative bacteria. However, once you add your decolorizer, whether it be alcohol or acetone, that does degrade and disrupt the outer membrane. So the outer membrane actually gets washed away at that point. And because this peptidoglycan is so thin and porous, the crystal violet at that point escapes. It can't be retained. In other words, there's nothing locking it into space here anymore. Okay. So it's not the outer membrane itself that's preventing the dye from getting in. It's actually the outer membrane is holding that dye in between those two layers. And then, once that crystal violet has been washed away, the saccharin can now bind into that peptidoglycan and form and bind into this lattice work here to prevent it, or to give that color, sorry. What else contributes to the ability of gram-positives to hold on to that stain? There's a couple of features here. One is the thickness, and that does have a big role with it. So that does help to kind of enhance that color a little bit. What did we say is the chemical that we add that actually helps to create these large complexes of crystal violet that lock it into that meshwork? The iodine will lock it in and actually make these complexes so large that they can't, it's as if they can't get out anymore. In alcohol, I know I mentioned this to at least one lab. Alcohol is good at dehydrating. What does that do to the peptidoglycan? It shrinks it. And that also helps to lock the crystal violet into place as well. Okay, so you've actually got a couple of things going on here that are contributing to this gram status overall. All right. Let's see if there's anything else I need to cover here. Any questions on just the basic structure or the gram staining and how that structure contributes to it? Yeah.
Can you say that when the decolorizer it removes the crystal violet, is the crystal violet in the periplasmic space or in the?
So it would be in the periplasmic space, but there's some of it bound up in that. Bacterial cells do have a cell wall. And so that inner membrane is going to expand, but that cell wall helps to prevent the expansion further. Okay, so that's how those cell walls can help to prevent osmotic lysis. Now, there are cases when this is not, or there are cases when this can be overcome. So, historical note. Before, I'm going to pull up the picture of gram-negative again here. So, before penicillin's activity was found in the 1920s, Fleming, who was one of three actually, who got the Nobel Prize for identifying penicillin, was working with an enzyme known as lysozyme. Lysis is in its name. Lysozyme we find pretty much, gosh, it's ubiquitous. And what I mean by that is it's in your tears. It's in your mouth right now. And lysozyme is an enzyme that hydrolyzes NAG and NAM. What does that mean? So when we say it hydrolyzes NAG and NAM, what do we mean there? So let's break it apart. Hydrolyze or hydrolysis, what kind of reaction is that? Going back to breaking down by adding water. And what does NAG and NAM refer to? Or N-acetylglucosamine and N-acetyluramic acid. It's the peptidoglycan. So these greens and browns here represent repeating units of the brown is NAM, the green is NAG. And the reason why I know that is because associated with the N-acetylmuramic acid are these tetrapeptides, or these 4 amino acids that help to hold those individual glycan strands together. So if lysozyme hydrolyzes NAG and NAM, what does that do to the cell wall? Yeah, it starts to break it down. So by adding water, in the case, I'm going to draw this on the gram-positive right now just to show this. What that allows lysozyme to do is to start breaking these bonds here. That reduces the integrity of the membrane. Now, if the cell's in a hypotonic solution, what happens to the cell at that point? It bursts. The cell wall becomes so weak that it can't repress that turgor pressure that's being built up inside of the cell. Okay, and when that happens, then the cell ends up exploding. First line of defense in the human body, lysozyme. Okay, it's one of the things that helps to protect us from potentially pathogenic microorganisms. Let's go back to the gram-negative form. Will lysozyme work on a gram-negative? We just talked about gram-positives, but will it work on a gram-negative? So if you have, say, E. coli and lysozyme and pure water, will those E. coli cells explode? I'll give you 2 minutes to talk it over with somebody else.
I think it would have to have a protein channel. You would? I think it would have to have a protein channel. They can't pass through free like do they? Can't pass through the outer membrane, so the lysozyme. Yeah. Right? I don't know, because I thought it was right? So it was like penetrated. Yeah, water can be easier, but you have to have the...
Yes. So if you have E. coli, which is a gram-negative, lysozymes up here, because this is outside of the cell, will those cells actually lysozyme? Meg, I have your lab assignment for Tuesday in my office. Feel free to come by and grab it if you want. The microscope one.
Okay.
All right. Who thinks they've got it? Claire has it. Claire, go ahead.
So I thought that because, what is it,
Lysozyme?
Lysozyme was a protein, it would have to have a protein channel to get into the membrane because it's too large to just pass freely through the outer membrane.
And there's no protein channel for lysozyme. So one of the things that we've talked about already is that selectively permeable outer membrane. And that it allows for hydrophobic molecules to get through, allows for nonpolar molecules to get through, but big, bulky molecules will not get through that outer membrane. In fact, I don't know of a porin large enough to fit another protein through. And so one of the failures of lysozyme is that it has no effect on a gram-negative cell if it's alone. If you were to add something like a detergent or alcohol as a step before adding the lysozyme, some way to disrupt that outer membrane, then yeah, lysozyme works. But if it's just lysozyme and just the cell, it has no effect whatsoever. All right, we'll finish with that for today. So, just a couple of quick notes before you all go. All right, so we are on structure and function number 3. This is the last, and actually next Wednesday represents actually just past the halfway point to the first exam. Just giving you a heads up, not to freak you out, just it's coming at some point. The assignment for this one is individual. It's a two-parter, and so let me take a minute to describe what that means. We've actually already covered some of this stuff in here. So it's a journal entry. So the only people that will see it are you and myself. So I want you to answer this question below. Typical concentration of NaCl in the cytoplasm of a human red blood cell is 0.9. What will happen if it's placed in a solution of 0.1% NaCl? Is the solution hypertonic, hypotonic, isotonic relative to the cytoplasm? Answer that one. The part that's in blue there, I'm going to have you answer after class on Wednesday. Okay? So I'll just have you go back and add that little bit to it, and that's the full credit. However, since we're almost halfway through, one of the things I kind of promised at the beginning of the semester was this is a new way for me to teach this course. So I want to get some feedback from you guys on how is it going personally for you right now. Okay? And there are no hurt feelings. If you're like, this sucks, I hate doing this, you know what? I will probably tell you the same thing I tell my children, but that's okay. Suck it up, Buttercup. I only tell Reed that. Jake's just a pain in the rear end. Anyway, so are you keeping up with what's being expected? So are you keeping up with the homework right now? Are you keeping up with the mini lectures? What's been beneficial for you personally? And then do you have any feedbacks and concerns about the course, the format, anything like that? This just stays between you and me, okay? And what I'll do is I'll take a look at it and then try to do what I can to address the concerns. The last thing I want to point out real quick, I have to say, from a personal standpoint, I've been really impressed so far with the amount of work that you guys have put into this and just how engaged you've been in the class. This is, honestly, last year was a tough year for me personally because we were moving and Jake was undergoing his transition and stuff. And so this year, in some ways, is like been a breath of fresh air. I just have to say that. I actually went home Wednesday smiling. And my wife asked what was wrong. She may have thought I was drinking, but in all honesty, I mean, honestly, the engagement that you've brought in, this is what keeps me coming in. I'm really excited about it, so thank you guys for your work so far. All right, with that said, be safe, have a great Labor Day weekend. I'll see you on Tuesday in lab. No homework or anything for Tuesday's lab.